Hello, everyone. So my name is Mike Reapy. As a UC Santa Cruz Alumni Council member, I'd like to welcome you to our monthly speaker series, Slugs and Steins, Lectures from UC Santa Cruz. For those who are new, our Slugs and Steins series engages a UC Santa Cruz faculty member in discussions with you, the local community of Silicon Valley and beyond our extended community online, with a goal of making all of us Renaissance women and men. We wanted to feel just like you're at UC Santa Cruz sitting in class, but with drinks and no exams. David, another volunteer organizer, is with me tonight. We're both alumni, and we spend our days in entrepreneurial companies, Silicon Valley style. He'll be helping me with the Q&A, and you'll hear more from him at the end. Before we get started, we'd like to take a poll to get to know our audience and where you are tuning in from this evening. So please take a moment. The poll should be up right now. Answer a few quick questions that will pop up on your screen. So we'll share the results after we've given everyone a few moments to respond. And I'll answer myself. And we'll pop up the results in a minute. You should be able to see the results of our poll and who is in the virtual room with you. So tonight, we raise a virtual Stein with Dr. Rebecca DuBois, Associate Professor of Biomolecular Engineering. Oh, my cat's attacking me. 1 second. the fun of Zoom working from home, right? So Associate Professor of Biomolecular Engineering at UC Santa Cruz. Her lab uses protein engineering to investigate human viruses and to develop new vaccines. Today is Rebecca's second talk for this series and comes just shy of 3 years since her first. She's actually the first repeat professor at Slugs and Steins. So mildly stated, events in the interim have moved her promising research to center stage. Rebecca received her PhD in biochemistry at UC San Diego and was a postdoctoral fellow at the Pasteur Institute in Paris, France, as well as St. Jude's Children's Research Hospital in Memphis. She lives in Santa Cruz with her husband, UCSC men's basketball coach, Ron DuBois, and their 2 children. As an aside, she has winemaking in her genome, but you'll have to ask in the Q&A for stories from her wine domain. We hope that comes up. We'll save questions for the end this time. If you have questions for our professor, please type them in the Zoom Q&A box down at the bottom. You don't need to wait until the last minute. You can type them at any time as they occur to you. If you see someone else's question that you like, you can upvote it and we'll ask it sooner. The talk is being recorded. In a few days, you'll be able to find it on the UC Santa Cruz Arts and Lectures YouTube channel. We'll post the link in our social media channels and follow-up emails. Okay, does everyone have their Stein? Great, I've got your slug, Professor DuBois.
Okay, hi everyone, cheers, welcome. Thank you so much for joining in today. I am so happy to be here and tell you about some of the work that we have done, looking at really shifting some of our research and branching off into a brand new research direction in response to the pandemic. As was stated in the introduction, I presented in this series 3 years ago. And this was my first slide. I said, how many viruses are in the news that you hear about lately? And people were naming off, you know, Ebola and Zika and some other ones, you know, bad flu season and talking about shingles vaccine. And I just have to point out that one of the stories that I posted was a report about how the next pandemic will likely be a respiratory-based virus. And, you know, this was the thoughts on a lot of scientists and public health experts saying we need more surveillance of viruses in the human population. So as of today, this is the virus in the news. Everybody knows that. And so for the last, you know, year, this virus has really taken over in many ways of our life. So when the coronavirus emerged, my lab asked, you know, how can we help and use our expertise to help with address this pandemic? And so today I'm going to tell you about our, give you a little bit of an overview and then tell you about what we did. So first I'm going to tell you about how our immune system responds to virus infection or vaccination, how the current COVID-19 vaccines work, and then I'm going to get into serological testing to detect antibodies in the blood. So how we do that and why we would want to do that. So to start off, I think everyone has really gotten Immunology 101 and Virology 101 in the last year. But to summarize, when someone becomes infected with SARS coronavirus 2, their body has an immune response, and part of that is the development of antibodies. These are little Y-shaped proteins that float around in our bloodstream. And so that's why they're in the shape of the Y. If we get a COVID vaccine, our immune system also responds by generating these Y-shaped proteins called antibodies. So how are these, getting back into the details a little bit more, how are these antibodies made and how are they protective? So we have a type of white blood cell in our blood called B cells, and these are antibody producing cells. So this is the cell here, and these are little antibodies on the surface. And when they happen to, when a specific kinds of antibodies are able to recognize a virus, they proliferate and they start making lots of antibodies and secreting them out into our blood. And when we have these antibodies in our blood, they are able to recognize any future infection and neutralize the virus. They stick like glue onto the surface of the virus and prevent it from being able to infect our cells. So antibodies are protective because they block future infection. So how do our vaccines work? And I will talk about the two main vaccines that are available in the United States right now. The first kind of vaccine is the mRNA vaccine. These are produced by Moderna and Pfizer. And so if we just look at the SARS coronavirus 2 virion, there are hundreds of genes in the virus genome. But the mRNA vaccine encodes only one virus gene, and that is the gene that encodes the SARS coronavirus spike glycoprotein, or just protein here. So when we receive an mRNA vaccine, that vaccine particle gets taken up and it tells our own body's cells to produce the coronavirus spike protein. Again, this is not infectious. It is just one protein being made by our cells. And these cells get displayed to our immune system, and that allows our immune system to respond and develop antibodies against that spike. You've also probably heard about the Johnson & Johnson vaccine and the Oxford AstraZeneca. These are adenovirus vectored vaccine. And so what that means is a harmless adenovirus that is engineered so it's non-replicating, is engineered to contain the SARS coronavirus spike gene. When we are immunized with this vaccine, the adenovirus is able to enter our cells and direct our cells to produce the SARS coronavirus spike protein on the surface of the cells. And much like the same way in the mRNA vaccine, that stimulates our immune system to recognize these spike proteins as foreign and develop antibodies against them. So how do we know these vaccines work? These are data from the phase 1 clinical trials showing the antibody responses to these vaccines. And I'll try not to give you guys too much data. But here is what you see. The height of the bars in either graph are the levels of antibodies to the SARS coronavirus spike protein. So you can see people that were never infected have these low levels or really nothing. People that had COVID-19 disease had various levels of antibodies after infection. And these are vaccinated individuals in the 18 to 55 and then 65 to 85. And what you see is you get antibodies after your first vaccine, and then you get your second dose, and you even get a boost in higher levels of antibodies. And these are known to last for months. And it was really great to see that these antibodies are stimulated in the 65 and plus individuals, because that's a really important age group to protect. We see the same kinds of increase in antibody levels to the Mrna vaccine, as well as the other ones. I'll just point out the word ELISA. That is the technique used, the serological test used to test for the antibodies in this. And I will tell you why that is a really challenging or a long, tedious experiment to do, and how we've tried to improve upon that in my lab. So these are the results of the phase 3 clinical trial. This is where participants, thousands, 30 to 40,000 in each group, either received placebo or the vaccine. They were double blinded. The participants and the doctors did not know which vaccine they received. And then all participants were monitored for COVID-19 symptoms. And what you see is those that received placebo showed COVID-19 disease over time, whereas individuals that received the vaccine showed a plateau and very little COVID-19 vaccine or COVID-19 disease after vaccination. And this is true for Pfizer and Moderna. And this has led to these two vaccines being FDA approved, and now the Johnson & Johnson vaccine being FDA approved. And I took this screenshot today. Over 119 million people in the United States have received at least one dose of vaccine. This is a massive accomplishment. We are at 36%, fully vaccinated at 22%. So I really feel like there's a light at the end of the tunnel. We are getting there. And I want to encourage everyone to sign up and get their vaccine as soon as they are eligible. So now I'm going to get into serological testing and tell you what my lab did. So serological testing is used to detect antibodies in the blood. When someone is infected with SARS coronavirus, they develop antibodies to a couple different proteins that the coronavirus makes. We've talked about antibodies against the spike protein, but people also develop antibodies against the nucleocapsid protein. That's this protein that covers the virus genome. And so when we were trying to decide how to develop our serological test, we had to decide which one did we want to use as the antigen, almost like as the bait to capture antibodies in the blood. And so what we reasoned is that, so the nucleocapsid, antibodies against the nucleocapsid show evidence of past infection, but they would not show evidence of past vaccination because we don't receive the nucleocapsid antigen in our vaccines. Also, antibodies that recognize the nucleocapsid do not block virus infection, so they are not neutralizing. On the other hand, antibodies against the spike would not only show evidence of past infection, but they also show evidence of vaccination. And these antibodies are known to be protective. They bind that spike protein on the surface of the virus and they prevent it from promoting infection. So I just want to point out that many of the serological tests on the market show evidence of past infection, if you're curious about that. But if you get the vaccine but never had COVID and you go get a serological test, it might be negative if it's testing for antibodies to the nucleocapsid. So if you want to see if your vaccine works, you would need a serological test that uses the spike. So that's what we chose to use in our serological test. And so the first COVID-19 antibody tests were two different kinds. These are really the gold standard of serological tests. One's called a lateral flow immunoassay. It's rapid and it's qualitative. So usually you take a couple drops of blood and put it on a little test strip, and then you get a positive line if you have antibodies against SARS coronavirus 2. But these are qualitative. Usually you get a yes, no stripe on these tests. However, they're fast. On the other hand, there's the ELISA. That's the test I mentioned was used to evaluate all the vaccines in clinical trials. This stands for Enzyme-Linked Immunosorbent Assay. You don't need to remember that. But what I will explain is that this is a really time-consuming experiment. It's tedious, but it can be semi-quantitative, meaning it can measure levels of antibodies in a person. You can imagine that low levels might not be protective, and there's some sort of threshold to where you can measure that protection. So what we sought to do is develop a rapid and semi-quantitative serological test. So let me go back to ELISA really quick. ELISA requires a lot of pipetting, and I will just state that it's multiple steps. You wash in between, and it can take several hours for different incubation steps. So usually this is about a 1-day long experiment to do ELISA, and that is the gold standard. So we asked, can we develop a faster way to measure COVID-19 antibodies? And we wondered if an instrument or technology that we use in the laboratory could be applied for this purpose. And that technology is called Layer Interferometry, BLI. And what this is a technology that uses really a fiber optic biosensor. These are biosensors that we can purchase, and an instrument will shine light down it, and that light reflects. And if there is a change on the surface of the biosensor, we get a change in the wavelength, and we can measure that change in wavelength as a signal. And so what's happening in our assay is we look for antibodies binding to the spike at the surface of this biosensor. What does this look like in real life? This is a picture of me in front of the Layer Interferometry instrument that we have on campus. And this is what a little biosensor looks like. And so what the instrument does is dip the biosensors into samples inside a little microplate here, and then it measures the signal that's happening on the surface of that sensor. So what we do is we put the antigen, in our case, we use the SARS coronavirus spike, into the assay plate. We put plasma, human plasma samples, and plasma is just a fancy word for the part of our blood that's not the blood cells. It's just the rest of the liquid. And then we have a detection reagent, and this looks for human antibodies. And I will explain that in a minute. So here's how the experiment goes. The instrument will dip a biosensor into a buffer just to get the steady state. And I'll just mention that we get real-time data. We get to watch the experiment as it happens. As the biosensors get dipped into the spike protein, we see a shift as the spike gets immobilized onto the biosensor. We do a quick wash, and then we dip into human blood plasma. If there are antibodies in that blood plasma, we see a signal as the antibodies begin to bind at that biosensor surface. We then wash again, and just as a confirmatory step that we find is the most sensitive part of our step, we do a detection step, and we use another antibody that's called an anti-human antibody, and that will recognize any human antibodies that are at this level and give another boost in signal. So really we get this real-time data, and we can measure really 2 different steps. And I will just point out, this whole experiment takes 18 minutes. It's just super efficient and really sensitive way to test for antibodies. When we wanted to develop this assay, we obtained a lot of pre-pandemic plasma samples through a collaboration with the Lyme Disease Biobank. And so I'm really grateful for their donations. We also obtained samples from a company that obtained plasma from people that had COVID. And so first we confirmed all of these samples using the gold standard ELISA. And just to remind you, it takes 24 hours to do this experiment. And what you'll see are what SN is called seronegative, all the pre-pandemic samples, and they're all quite low in signal by ELISA. And then the COVID positive patient samples are in red here. And so we see a signal. So we've confirmed by ELISA that these samples were positive. There was one that was quite low, but in general, most of them were detectable by ELISA. And here's what we see with our assay. Here's a seronegative and a seropositive. And so we would measure wash, we see the spike binding to the sensor, and we see the plasma incubation step. And a seropositive individual will get an increase, seronegative, nothing. And then finally, our detection step confirms seropositive, whereas a seronegative person would have a flat line here. And again, we can quantify that increase, and that shows it here. And what we find is that our serological test is very sensitive and is able to distinguish all the pre-pandemic seronegative samples here, and also detect all 10 seropositive samples, including that one sample that was very weakly seropositive. So we're really excited to have developed this test. We show that these levels are very similar to ELISA levels. And so that I think is going to be important in the future, understanding what that level is. That means you're protected if you're above this level. We published this data in December, and we're really excited to share this information with the world. I do think this is really could be a technology that takes over and gets rid of ELISA that is just such a tedious method. And it's really hopefully an advancement for the community. I'll say we've tested, since we published, we've tested a lot more samples, hundreds really of samples, and really confirm all the seropositive ones show up and all the pre-pandemic ones are negative. And so we're really excited to further validate our serological test. We also did a relatively small serosurveillance study. This was in July last year, but we had individuals self-collect a few drops of blood. After centrifuging, we transferred the plasma into a little assay plate here, and we asked how many people were had SARS coronavirus 2 antibodies in their plasma. And so this is, again, this is July. I'll just show you here the detection step, but we only had about 2 or maybe a few on the borderline here. So a very small number of people were seropositive at this stage in Santa Cruz, but it was a great way to just further use our test. And finally, this is really recent data, but we've gotten samples from an individual that received a vaccine, and they self-collected samples using this method. And what we find is that we really see, so this is day 1 of week 1 of the vaccine, and about 2 weeks, we see detectable levels of antibodies. And I think the second dose happens on week 3, and we see an increase, a further boost in antibodies over time. So it's a great way to confirm that the vaccination works. So in the future, we're hoping to take the data and apply for FDA EUA emergency use authorization approval of this serological test for use on our campus. We, in my lab, we're often making antigens to a lot of different viruses. That's a big part of what we do, is study the antigens and the molecular structure of them. But we really could expand this technology, and we have started to for one other virus, and we show that we can detect antibodies to other virus antigens in those same plasma samples. And I'll also mention that I am currently teaching one of the first in-person classes of this entire academic year. I fought very hard to have it taught in person. protein engineering lab. And what these students are doing is learning how to make the SARS coronavirus 2 spike protein in the lab, and we'll use that for the serological test. So it's really exciting and fun to teach the next generation of students. So finally, I will thank all the wonderful lab members that contributed to this project, especially John, who was the first author on that paper, really worked hard to help push this out, especially when we really had to social distance in the lab. Again, I just love my job and that I feel like I'm trying to inspire that next generation of scientists. And this is a picture that a former undergraduate student from my lab sent to me, and he got a virus tattoo on his leg. So I just, I love that enthusiasm of the students that come through my lab and my classes. And finally, I want to thank UC Santa Cruz and Lyme Disease Biobank for their support, as well as a lot of this research was funded through the Michael David Family Foundation. I will say that Michael's my father and David's my uncle and grandfather. And through the Michael David Winery, proceeds from the Going Viral Merlot went to support the research in our lab. And if you'd like to support the research, please go here and donate to the lab. So thank you again for listening, and I'd be very happy to take your questions.
Thank you so much, Professor. So actually, you started in on the wine story. Is there a connection between viruses and wines?
Is there a question? Say it again.
A personal connection with you, viruses and wines. What is that?
Yes. I have the fortune of coming from a wine family of grape growers and winemakers. And so I am a little bit of the black sheep of the family to not be in the wine business, but it's really great having that. And I think that my backup job would be maybe working in the winery or perhaps studying viruses that infect grapevine plants.
Okay. So we have Jack asking, does the vaccine produce antibodies for immune suppressed, for example, cancer individuals?
That's a really great and important question. Those individuals were not included in the phase 3 clinical trials. And now many of those individuals are being evaluated for that. And my understanding right now is the answer is it definitely depends on the type of immunosuppression. But there are some studies coming out now that show that some immune suppressed individuals do not respond as well to vaccination. But I think it definitely depends on the type of immune suppression that you have.
Okay. We have a question from Catherine. Can COVID-19 antibodies present prior to vaccination be detected and distinguished from the new ones?
I think I'm not fully understanding the question, but maybe if you had COVID-19 disease and then you got the vaccine, that's where the serological testing, the specific type of test could distinguish. So if you got a test that looks for nucleocapsid, antibodies against the nucleocapsid, those will only be induced by natural infection. So if you really want to know that if you had natural infection before you got the vaccine, you would have to get that very specific kind of serological test. The ones that evaluate the spike will not be able to distinguish between natural infection and vaccination.
Question from Hugh. Are biolayers developed for each protein or biomolecule being detected? And if so, how robust are the biolayers to repeated use and tests?
So these biosensors are, in the way we're doing it, are single use. They can be sort of washed and stripped away, and so you could reuse them for different proteins or different antigens. I do have a big picture goal. Imagine you could donate a blood sample or give a blood sample to your doctor, and we could use this method and dip, 20 biosensors into your plasma, where every single biosensor has a different antigen on it. And so they could say, all right, you are protected against flu and pneumococcal infection, but you don't have strong enough levels to tetanus. We want to give you a vaccine and boost your antibodies to that. So that's where I think is a really exciting potential for this technology.
Question from Liana. Which lasts longer, natural antibodies or vaccine antibodies?
Ooh, good question. I am excited to hear what the answer is. Right now, we know that antibodies last for at least 6 months. I think we're out to about 8 months now, really confirmed. What we do know is that natural infection to other coronaviruses, the ones that cause the common cold, that they wane over time. And so we can be reinfected with the same exact strain of a common cold causing coronavirus, a few years later. And so the question is, will antibodies from, natural infection, will they wane over time in that same way, or will they persist? And same with vaccination. We have we're learning a lot about mRNA vaccines, but we don't really know the full longevity yet about those antibodies.
Okay, this is from Kevin. Which cells take nanoparticles? All of them or just specific ones? Does it matter?
Right. I missed it a little bit, but I think what kind of cells take up the mRNA vaccine or any of the vaccines? Was that the question?
Well, it was described as nanoparticles, so I think I would guess you're right.
Okay, so I think referring to the mRNA vaccines, it's just believed to be the muscular cells in your body, I think mainly that are sort of transfected. The nanoparticles get into the cells, but I think it really is any cells in that area. Uptake the mRNA and then produce the spike protein.
Question from Jorge. What was one of the biggest difficulties in coming up with this method, and how did you and your team overcome
it? Ooh, good question. I think probably one of the biggest challenges was getting our hands on pre-pandemic plasma samples. Everybody wanted those because they were definitely would not have antibodies, and that's an important sample for validating your assay, being sure you have no false positives, right? So part of it was just getting access to those important plasma samples. And we just kept asking colleagues. But another thing was figuring out different kind of dilution buffers and things, because plasma, as you can imagine, is a very complex liquid. It's got all sorts of proteins and hormones and small molecules and things. And so how do we make sure that any signal that we observe from plasma is not some false positive? And so we had to really sort of experiment with different solutions to make sure that doesn't happen.
Bill asks, how do the antibody levels look over time?
Right. So we're not sure, beyond say 8 months, which is when all the clinical trials have started. But in general, they really peak after a certain few weeks after your second dose, and they wane a little bit, but really they stay strong for months. That's as much as we know so far.
From Donna. As someone deficient in IgM, immunoglobulin M, would I be expected to have a normal vaccine response?
That is an interesting question. IgM is a type of antibody that really is sort of your first antibody response to a vaccine or infection. And then as the antibodies mature, they develop into IgGs. So I'm not entirely sure about the mechanism of that deficiency and whether that would affect it, but I would love to test your blood.
Okay, Christian asks, if I understand correctly, the ends of the biosensors will need to have a binding molecule in order to bind the antibodies. How long and through what preparations does it take to bind the biomolecules to the ends of the biosensors?
Great question. That's really a protein engineering strategy that we used. There are little amino acid tags that we can encode at the end of the proteins, the spike protein that we make in the lab. We also added a few extra amino acids, and that allowed us to put a small molecule called biotin tag to it, and that basically made it permanently attached to the surface of the biosensor. So a little bit of protein engineering in order to get that to work.
From Jamie, if the vaccines have inactive ingredients that are dangerous to an individual and that person has antibodies, could they be protected from the virus without getting vaccinated?
So people should look at the ingredients on a vaccine and see if they have known allergies to any of those ingredients. I think sort of this relates a little bit to another question, is natural infection antibodies versus vaccination. And so the clinical trials actually show that natural infection really has a little bit more of variability from person to person in terms of their antibody response, compared to vaccination, where everybody really gets this strong response. The SARS coronavirus genome really has 100 genes in it, and most of them are meant to thwart our immune response to block the virus. And so some of that sort of molecular warfare, I think, by the coronavirus prevents a really strong antibody response in some people. So I think the recommendation is that people that had COVID should still get the vaccine to boost their antibody response to that level that they know is protective.
Manuela asks, in one of the slides, it looks like antibodies dropped by week 6. Am I reading it right?
Yes, let's see. There was a little bit of dip in week 6, and that's a natural thing. You really get this sort of boost and peak in antibody levels, and then it really sort of levels off after that. So you might get a slight decrease, but it's not something that's going away. We know that these antibodies last, these levels last for a long time.
Question from Janet. Can your method detect antibodies to virus variants? So I guess that means, you could discriminate between different versions.
Right. That's a really good question. And I think that's something that people are going to be looking at more. I think the real answer is going to be, it depends. It kind of depends on the individual probably and the specific variant that you're referring to. In general, people develop antibodies and they stick to all sorts of different parts of the spike. It's not just like one spot. And if there's a variation, that might prevent some antibodies from binding, but still you're going to have antibodies binding to all the different spots. So for the most part, I think the methods will still work even if you were exposed to the variant.
So Anita puzzles about the difference in age groups. Do you know why older people who get the vaccine seem to have less or mild or no side effects versus younger groups?
I know, I wonder that too. I feel like my grandma didn't have the vaccine didn't phase her or anything, but then a lot of people in my lab were out for a day because they weren't feeling great from the vaccine. So I don't know if that's anecdotal or really officially something about the vaccine. We do know that as the older population doesn't have as strong immune responses to any kinds of vaccines. The flu vaccine just doesn't work as well in older individuals. And so it's probably something related to age and sort of a not as robust immunity when you get older.
Question from Kathy. We'll see if this is in your wheelhouse. How does a vaccine become approved and not just have an emergency approval?
Oh, that's a good question, and I should know that. I'm sorry, I don't think I have an official answer, but I do think that I think that is coming down the pipeline, is full approval of our vaccines. I imagine there's this window of safety evaluation, longer term safety evaluations during the EUA emergency approval period, but I don't know exactly the timeline on that.
Christian notes that he's been told that the reaction to the second dose is more severe than the reaction to the first time. Can you confirm that? Why is that?
Sure. I think it might be anecdotal or it might be true, but I do think part of it is you are re-stimulating your immune response. So it's not just your first response, it's sort of reactivating, and you already have existing immunity from the first dose. So it's sort of like a double stimulation to get your immune system going. And that second dose is actually, it's really important. It helps your antibodies become tighter binding, they work better, you get higher levels of antibodies. I showed the data for that you really do get higher levels from that second dose. So don't shy away, do get your second dose.
Okay, you're really moving through the questions, but keep these coming, everybody. From Jordan, could you comment on mRNA vaccines moving forward? Are they more effective or better than traditional attenuated protein subunit vaccines? And if so, why? What about from perspective of industrial production? Does the instability of mRNA prove problematic?
Oh, that's there's a lot of parts to that question. You know, it's actually, you know, it's a benefit from this pandemic, is that it is given the need to test different kinds of vaccine technologies, and mRNA vaccines work really great. I mean, we were just so excited about really the efficacy and how robust they are. Are they more effective? They might be. If we converted our flu shot to an mRNA vaccine, would it work better than our traditional made vaccines? I'm sure that's being tested. I can imagine a vaccine in the future that's flu plus COVID protection and potentially others. So I think there's a lot of potential with mRNA vaccines. Are they going to completely take over other vaccines? I don't know. We obviously know they have major cold chain issues. Cold chain meaning they require freezers, and that is really challenging for a big part of our world. And so I think a lot of different types of vaccines are still going to be important moving forward, and not everything's going to switch to mRNA.
I also asked about the production. Are they more difficult to produce?
Yes. Well, no, sorry. They actually can be produced. You know, a lot of the technology is a little bit, you know, not fully transparent right now. But so mRNA is not very stable. That's why it has to be encapsulated in these little nanoparticles and kept frozen. But it's a really fast and quick way to make a vaccine. So we are so much more prepared for that next pandemic, because we can design mRNA the day that we get the sequence of a new virus strain or pandemic, and that can go into a pipeline much quicker than a lot of the more traditional vaccine technologies.
Kevin asks, is anyone biobanking plasma samples for negative samples for the next pandemic?
That is a great idea. We need to be doing that, because as I said, that was one of the biggest challenges that I had, was really connecting with a biobank and getting those samples. I will say, yes, go donate your blood. Many of these places will pay you for your blood, and they're really important and valuable research reagents.
From Jack, do more side effects to vaccine correlate to increased antibody response?
I don't know. That's a good question. We do know that most people develop a very robust antibody response, but then the side effects from the vaccines really vary. So I wouldn't guess that, but I'm not sure.
Selena asks, in a recent study at Cedars-Sinai, people who've had COVID-19 mounted an immune response after one Pfizer BioNTech vaccine equal to people who hadn't had COVID-19. So the second set, after they had had the second vaccine. Do you think this should change the protocol over time and save on those vaccines?
That's a great question. I thought this would actually change, protocol faster, but it hasn't become an official protocol in terms of saving on vaccines. I'm not sure why I assume that would require an actual extra clinical trial to test for efficacy of someone that had COVID plus only one shot. But in terms of antibody levels, the immune response, as you can imagine, 2 doses of vaccine, you give just as high levels as someone that had their first dose was an actual infection and then a second dose of the vaccine. But I don't know if that protocol will change. I'm not sure.
From Regina, did one of your charts show a decline of antibodies 6 months after full vaccination?
No, I
Do we have
think
any data on
I think you're referring to this one. This is just one individual measuring the antibody levels weeks after the dose. And so these are probably all pretty similar. There might be a slight dip, and as I was saying, that's normal to sort of have a peak, and then it would sort of most likely level off after this time. And we do know that those vaccine levels stay pretty high over time, months after vaccination.
Jack mentions that he's getting an antibody test at LabCorp.
Hmm.
Will it be the yes, no, or a quantitative test?
I am not sure. That's a good question. I would say the majority of tests on the market are yes, no. Also, the question is whether it's going to measure nucleocapsid or spike antibodies, and what you're trying to answer with that. If you got the vaccine but never had COVID, and you're getting an antibody test against the nucleocapsid, you're going to be negative. But that doesn't mean your vaccine didn't work. It just meant that it wasn't the right serological test to measure antibodies against the spike. So I'm not sure. It's probably going to be a yes or no.
From Mitch, when will a booster vaccine be on the market? Will we have to get these vaccines every year now, and how long will the vaccine last? So maybe another...
Great question. I do know booster vaccines are being developed with at least one or more of the variants that are starting to emerge. Whether those are needed, when they'll be needed is still to be determined. Will we have to get these vaccines every year? Is it going to be like a flu shot? It's not clear. We do know that coronavirus doesn't mutate as fast as the flu does. It's a different way in terms of where it mutates or how often it can mutate. But there's just so much coronavirus infection right now that's why we're seeing mutations happen. It's not that it mutates more, but there's so many more infections in our world right now that the chance of getting variants is higher. So I do think that a vaccine will probably protect for, I don't know, a couple years, but to be determined. Again, we don't know the longevity of mRNA vaccines in terms of how long they'll be protective for.
Jean asks, was there any difference in post-vaccine antibody production depending on which vaccine was administered?
The Pfizer and Moderna, they used slightly different ELISA tests. So they're sort of apples to oranges in terms of the signals that they got. But I think in general, most of them induce high levels, robust levels of antibodies that are probably very similar levels. And I'll say anecdotally, we've been looking at that in the lab and we don't see differences between the Moderna and Pfizer vaccine levels.
Question from Dan. Is it possible to go back in time and figure out if a person had ever had COVID after they got the vaccine? Or can you not discriminate?
Yes. So if you get a serological test that looks for antibodies to the nucleocapsid, that's the antigen that's found that stimulates antibodies by natural infection, but you won't get those from the vaccine. You could get that test and see if you had COVID in the past and just it wasn't confirmed.
David asks, are you pursuing any opportunities to license or otherwise commercialize your technology for broader use?
Yes, we are. I am hoping to really prove that this would work in a lot of different scenarios. And we have written up intellectual property paperwork. We've submitted that to potentially license and commercialize this technology. Perhaps it could be a standard thing that is in clinical laboratories in, all around the country.
So question from Anita, can the Santa Cruz community get a rapid test for antibodies, I guess using your technique? And if so, how?
So we have approval to collect blood samples from people and evaluate them in the lab, but we do not have approval to give people their results back. That is not allowed. So we're allowed to study them. But part of the reason why we're applying for FDA approval of our test is so that it could be put to use on our campus in a clinical capacity and return results to people. However, there are clinical labs in the Santa Cruz community that do serological testing and to see if you have antibodies.
Ben asks, how much can you play with the duration of the antibody binding step in your assay? Could you shorten the length of this step to speed up the test even more?
Ben, you are on top of it. We felt like we really sort of played, that was another thing we played with, was the times at each step. We also played with sort of the concentration of our detection reagent, the detection antibody, to see higher concentrations would give a bigger signal. So we could probably shorten it to about, maybe 10 minutes total. Another thing we could do, we found that we could pre-coat the biosensors with the spike protein like the day before, and it would be perfectly fine. So theoretically, you could get all the biosensors pre-coated and ready, and then you would just have the plasma detection step that would only be, that would shorten it by a few minutes as well.
From Jose, do we have any data that supports news on pregnant women getting vaccinated and the baby getting antibodies?
Yes. There are now studies coming out that the vaccine is safe in pregnant women, and they do develop antibodies, and those antibodies do transfer to the babies, just like all the mother's antibodies. But that is a decision that a pregnant woman should have with their doctor to decide whether to get the vaccine while they're pregnant.
Patty asks, how did you get your antibody samples? Were you able to ask and get volunteers?
Yeah, that was sort of a little bit of a side thing. We were hoping to do a surveillance study too. I mentioned a sort of small 100-person surveillance that was not random at all. It was just colleagues. We asked them to donate samples. So most of the antibody samples we received from the biobank or from a company that was getting plasma samples from individuals that had COVID. But we did have that serosurveillance, and we developed a protocol taking a little lancet, that's a little retractable needle, to make a little drop in your finger and put a couple drops of blood in a tube, and those were donated to my lab. So we did evaluate about, 100 or so samples with that method. There were definitely varying results in terms of people and their enjoyment of collecting their own blood. I will say that I'm not a fan. I'm not a fan of needles, but I've overcome that, and I have, stuck my own finger dozens of times now to collect my own blood as a negative control, because I never got COVID and I knew I was antibody negative.
From Karina. This is really exciting work. Thank you for presenting today.
Yeah.
How well did your charts correlate with the ELISA results from the samples obtained from individuals who had had COVID? In other words, how closely is the quantitative part of your procedure coming together?
Yeah, great question. If I didn't show it, I just felt like it was getting to be too much data, but we really see perfect correlation between ELISA and our study, and that was really important. We didn't necessarily see it with this other test that measures total antibody levels. That was a little bit off, but the ELISA, which really is the gold standard, and ours it just matched perfectly, which was really exciting.
So we have a donor wannabe. Donna wants to donate her blood. Can she?
I could take blood, but I could not give Donna her results back. And so I'm not sure how much fun that would be, but we're really, I think at this point, like I said, we are, I'm teaching this to students in my lab class right now, and but my laboratory is sort of pivoting and trying to expand this technology to away from COVID and now into measuring antibodies against totally different viruses, the flu and RSV and some other respiratory viruses.
If I'm not mistaken, Donna was the questioner with the IgA. I think you said that you wanted to study her blood, right?
Right.
I don't know if you're local, Donna. Yeah, we'll introduce you. Another question from Jorge. It's popular on social media to say that one brand of the vaccine is better than the other. Do these claims hold any value?
Nope. The best vaccine is the one you can get sooner.
I think we're waiting for another question. I have one. You were talking a little bit about side effects from the vaccine, and I'm wondering, I think I heard there are ingredients that they put in the vaccine, they call them adjuvants, I think, that are sometimes put in there specifically to, to increase the immune response. Are there adjuvants in these vaccines?
There are parts of the nanoparticles that are that help to stimulate the immune response a little bit, but there are not your traditional. Oh, I should know this. They're not your traditional adjuvants, I don't think, in these vaccines. But I should probably know that.
So Dan wants to talk. Go ahead, Mike.
Oh, I said maybe she can follow up later. Well.
Dan wants to target the common cold. So can it's used for the current COVID-19 vaccines, mRNA and the other one, be usable as a vaccine against the common cold? Same techniques?
Ooh, yes. So now that we know how to make a coronavirus vaccine, we certainly could. And you know, sort of going even beyond the common cold, I think there really is potential to make what's called a pan coronavirus or a universal coronavirus vaccine that, you know, potentially would elicit immune responses against many different strains of coronaviruses, some that cause the common cold, some that cause COVID or SARS, but also potentially protection against other coronaviruses that might emerge in the future from animal reservoirs. So the potential to have immune responses that would prevent future pandemics is really exciting too.
Question from Regina about ingredients. Are there any ingredients in the flu shot that are also in any of the COVID vaccines?
I am not sure about that. I did just find the slide here on the mRNA vaccine ingredients. So it's the mRNA. These are lipids that make the little nanoparticle around the mRNA, and then salts and sugars and buffers to keep it at the right pH. But I don't think there's anything that's technically adjuvants, as we were talking about. But there are some things, you could see polyethylene glycol, that's sort of part of the lipid particles that might be found in some other things that might stimulate some immune response, but it's not totally sure. So I think might be asking if you had some adverse reaction to the flu shot, does that mean you will have an adverse reaction to the COVID vaccine? And I think that might depend on what kind of flu shot you got and also what kind of COVID vaccine you might get. So I would recommend you work that out with your doctor.
Leon asked, is one dose of vaccine enough to boost the immunity for someone with the natural antibodies?
Right, so there are studies like one out of Mount Sinai suggesting that someone that already had COVID-19, that one dose of vaccine would stimulate antibody levels to the same as someone that was naive and got 2 shots of the vaccine. So there's definitely evidence for that, but right now it's not an official recommendation. The official recommendation is to get 2 doses, even if you had COVID in the past.
Question from Bill, interesting. Would there be a benefit to getting both Pfizer or Moderna and a J&J vaccine?
That's a question I don't think I have the answer for yet. There probably are slight differences. I will say that all 3 of them encode the same exact protein, the spike protein. So your immune response, your antibody response should be pretty much exactly the same, no matter what vaccine you get. However, there are side branches of our immune response based on T cell immunity, and those might be slightly different between different vaccines. And so I think the jury's still out about whether that would be, there would be any benefit to that.
Richard wonders about the future and whether COVID-19 should be included in flu shots, so double dose vaccines. Would that make sense?
Yes, I hope so. The fewer shots, the better in my mind. But yes, I hope so. I do think that is the low-hanging fruit, is convert. See if flu vaccines, mRNA vaccines can be used for our flu shots, and mix that in with the COVID spike and have a multivalent vaccine for COVID and flu. But I hope so, but to be determined still, I think, especially for this fall.
Okay, well, with that, my goodness, you not only have the record for the number of Slugs and Stein lectures, but I believe you have the record of questions answered. So congratulations, Professor.
Oh, thanks so much. Hopefully I got them mostly correct there.
Well,
yeah, thanks
very
for
appreciative. Before we let her go, She had put up a link, and I'd like that link to be put up again and maybe slid into somewhere, sent out so people could find it. Because if you were moved by her work, and I would say also the students' work on this, please take the opportunity to let her support those students and make further important contributions. So again, thank you, Professor. I think that link will be coming up shortly. A big round of applause in the Q&A box for our Professor Rebecca DuBois. Thank you for sharing this fascinating research with us this evening. As mentioned, the talk was recorded and it will be available on the UC Santa Cruz Arts and Lecture YouTube channel in a few days. Also like to have a big round of thanks for the staff of the Alumni and Special Events offices who set this forum up and support it. Thank you, Diana, Paulina, Kristen. A super special thanks tonight to Cara Snyder, who after a couple dozens of these events where she has given us marvelous support, is leaving. We will miss her. She's going off to another fine institution, though, Berkeley. So thank you, Cara, and our best to you going forward. Our next event will be Monday, May 10th. We'll feature another than former UCSC Chancellor George Blumenthal, former Chancellor George Blumenthal, speaking on a century of paradigm shifts in our understanding of the universe. A theoretical astrophysicist, Chancellor Blumenthal made path-breaking contributions to our understanding of the origin of the structure in the universe, including galaxies and clusters of galaxies, and to the role of dark matter that it plays in the formation and evolution of this structure. He is the co-author of 2 textbooks, 21st Century Astronomy and Understanding Our Universe. George holds a BS in physics from the University of Wisconsin, Milwaukee, and a PhD in physics from UC San Diego. Now, meanwhile, and this is important, Alumni Week 2021 at UCSC is just around the corner, running from the 19th to the 25th. It will include dozens, many dozens of great events. The Alumni Weekend of years past has been reimagined as a full week of virtual events that you can pick and choose from, such as a live reading of Shakespeare's Comedy of Errors, a conversation with Terry McCullough, Oaks 91 Politics, who is now Chief of Staff to Nancy Pelosi, or a virtual visit with elephant seals at Año Nuevo. You can even learn from our own Mike Reapy on how to make kombucha. Trust me, more than one of these events is right up your alley, and you'll kick yourself later if you miss them. You can read more at alumniweekend.ucsc.edu. So on behalf of UC Santa Cruz Alumni Association, thank you for joining us. And please come back on May 10th, 6:30 p.m. as always, for our next virtual event. Thank you again.